everyone, Jai Hind and welcome once again to the platform of SRIRAM's IS. Now today we'll be talking about in this session about the gene edited rice and as well as GM crops and the promise, perils as well as the policy that is related to these different crops which had been introduced. Alright, now why this topic is important for today, alright, or why we have brought down this topic for you to understand this all in this today's session. It is because over here you can see that this is the news that was recently in news just a day before in Down to Earth, and that is the center claims about gene edited rice varieties don't match up analysis of ICR data shows. Alright, so it was on May 4th that there were two varieties of the genome edited rice that were, they were given and released by Union Minister of Agriculture and Farmers Welfare, Shivraj Singh Chauhan. Alright, these two genome edited rice varieties are DRR Rice 100, that is also known as Kamla, and Pusa DST Rice 1. And the major aim over here for introducing these rice was to increase the production as well as for the drought and salinity tolerance. These rice varieties had been introduced. Now, why this topic matters for India and why this topic matters for UPSC as well? It matters because when we talk about India, India is the largest. Now, please remember this, India is the largest producer of rice. According to the latest estimates which are available, now India has surpassed China. Earlier, China was the largest producer of rice, but now we are the largest producer of rice as well as one of the leading exporters all over the globe. So because of this, India is the largest or leading global rice producer. This is very important because the varieties, the GM varieties or the genome editing varieties that we are talking about today are both, they are of the rice crop. Alright, then when we are talking about feeding of 1.4 billion people in India, alright, so rice is a very prominent staple food crop, and then again it holds very importance for the food security in India. Also, when we are talking about the climate resilience, we have spoken about how the climate change has been, that is leading to a tremendous effect on various things that we are seeing, whether it is the weather, whether it is the agriculture, whether it is the landscape surrounding us. And when it comes to the agriculture, we need climate resilient crops which could be grown easily and they could tolerate and they can withstand the different perils of the weather that are there because of the climate change. So when we are talking about the adapting to the changing weather patterns, these genome edited or the genetically modified crops becomes, or GM crops becomes very, very important. Alright, also they enhance the farmer's income because they lead to increase in productivity, and when the productivity increases, we have seen this in the case of the Green Revolution. So when the productivity increases, ultimately it leads to the increase in the farmer's income as well. So therefore, when we are talking about the gene technology or the GM crops, they hold both pros and cons. Everything has their advantages and disadvantages, and therefore when this gene technology enters into the agriculture domain, over here we have to see it critically. Alright, we have to see both the things, that is pros as well as cons of this technology. Now when we talk about this technology, there are two different methods how the genes could be edited and we could have the genetically modified crops, and this is by genetically engineering or by genetically modifying it. Now when we are talking about modification or genetic engineering, now what happens over here is alright, what happens over here is that over here when we are talking about the genetically modified crops, over here what we do is we insert a foreign DNA. Alright, we insert a foreign DNA, and this DNA could be inserted, or this DNA is inserted basically from another species. We have another species, and from this another species, suppose we have a prominent gene that is related to a particular quality that we want to bring into another species of or another variety. So we will extract that gene, and then that sequence of the genome will be inserted into the another species, and that is how we have the genetically modified crops which are, they are produced. So over here we have cross gene transfer, cross species gene transfer, and we use traditional transgenics and over here the regulatory framework is very strict as it comes for the genetically modified crops. So you have to remember, when we are talking about GM crops, there is foreign DNA that is inserted and there is cross species genes that are there in a particular plant. Like for example, we have, or you must have read about BT cotton. Alright, when we are talking about BT cotton, so over here the gene has been taken from Bacillus thuringiensis. Alright, from Bacillus thuringiensis, we have taken the genes and inserted it into the cotton genome, and that is how the BT cotton was made. But when we are talking about genome edited crops, GE crops. Now what are GE crops? Here we have precise DNA modification. We use SDN1, SDN2 techniques, alright, nuclease techniques that are used, and there is just what we do is that there is a DNA. That DNA is cut and the faulty gene is actually restored, or the faulty gene is actually repaired by the different methodologies. We have SDN, SDN2, alright. So with these methodologies, the repaired gene has been, it gets cured, and then we introduce that genome into the crop variety, and that is how we have the genome edited crops which are made. Now, as you can see over here, there are no foreign DNA that is inserted. Only the faulty genes, the faulty genome strands are basically repaired by this method. So over here we have the exact genome of the species that was there before. So over here we have lighter regulation pathway, and there are precise cuts to the DNA that are made, and also the existing genes are edited by this method. So that is the difference between genetically modified and genetically or genome edited crops. Alright, so over here you can understand this, that where we are, interbreeding two different varieties of crops in the field. So over here what happens is that we have a donor variety, alright, we have recipient variety. So over here we get a new variety by conventional breeding. But at the same time, when we are, this is where, the success rate would be very less, alright. The success rates would be very less, but when we are talking about the genetically engineered or genetically modified crops or the genome edited crops, so what happens over here is, you can see over here very clearly that here what happens is that we have a donor variety. Like for example, I told you Bacillus thuringiensis. Alright, so over here in Bacillus thuringiensis, we had a gene of interest. We have taken this gene, and then this gene of interest was isolated. Alright, this was isolated, and we have a recipient variety which we had cotton. Alright, so over here in cotton the disease resistance was low and the yield was high, but we want the cotton to be disease resistant as well. So we have taken the gene of interest that is related to disease resistance and increasing the resistance of the diseases, and this had been added to the genome of cotton. And now we have a new variety where the desired gene is transferred to the location in the recipient genome. Alright, now what is genome editing? In genome editing, we identify the susceptible gene. The susceptible gene is that gene which is a faulty gene that is related to the decrease. Like for example, we have taken over here an example, disease resistance. So this susceptible gene is related to the low quality of the resistance related to diseases in this particular crop. Alright, now this genome is edited. This genome is edited by CRISPR technology and by using SDN1, SDN2 technologies, alright, and by using these technologies, this susceptibility gene is disrupted and the genome is edited, and again we have a new variety where disruptive disease susceptibility gene has been gone. Alright, so this is how both these technologies they work, or they are different from each other. Now we have the first, India's first GE rice varieties. These two varieties have been developed. We have the variety that has been developed by ICAR, that is called as DRR Dhan 100 or Kamla, which is basically developed for drought tolerance, and it targets the rain-fed rice growing regions that are there. We have Pusa DST Rice 1 as well, which had been crafted or created for direct seeded rice system and addresses the water scarcity as challenges which are there. Again, when we are talking about these two varieties, it is very important to remember here about the Green Revolution. Alright, when we talk about the Green Revolution has led to the regional disparities. Although development of these two varieties are very much favorable for India. Why favorable for India? Because when we talk about India, all right, so the green revolution's area has been basically spreaded over this particular region. All right, where we have over here, we have Punjab, we have Haryana, all right, we have Uttar Pradesh, and when we talk about Uttar Pradesh, only western Uttar Pradesh, and we have regions of Maharashtra which have reaped the benefits of the green revolution. But the other regions, they have not reaped the benefits of the green revolution. Why? Because when we are talking about green revolution is based on basically three principal SFI: seed, fertilizers, and irrigation. So these regions are basically the regions which are irrigation fed regions. There is ample amount of irrigation capabilities, and that is why these regions have thrived and they have done well under the green revolution phase 1. But now we are talking about, we have been speaking about the food security. We have been talking about the green revolution phase 2, and for that green revolution phase 2, we have to look for either the the crops which can be grown in drought like conditions, or where we have the water scarcity. And for these regions, if we have the kind of the crops that could be grown and that could sustain the crops could be a food crop, then it will lead to much better food security in the upcoming years. So that is why when we are talking about these rice varieties, the genome edited rice varieties, both the varieties, they hold very prominence for the green revolution phase 2 that is about to be launched in India. Alright, now what are the claim benefits of these GE varieties? So when we talk about the claim benefits, 19% of the yield will increase that had been promised over here, 19%. All right, so there would be higher productivity per hectare, and when there would be higher productivity per hectare, that will again lead to much better farmers' income. All right, the farmers' income will get boosted. All right. Along with this, when we are talking about the maturity cycle, it is 20 days earlier. That means a shorter growing duration for these rice varieties, as well as the most important thing is that there will be reduction of the irrigation needs, and 30% of the water saving will be done by using these varieties. These benefits have been claimed while introducing these two varieties. Also, so they are drought and salinity resilient or tolerant varieties. All right, they require lower fertilizer requirement. Again, that will have an impact on the farmer's income again, because they have to pay less for the fertilizers, and also that will lead to reduced methane emission. Again, when we talk about the rice fields are basically the source of methane emission, and methane is, again, it is a very prominent greenhouse gas that is there in the atmosphere, which leads to the climate change again. So of course, if it is leading to reduction in the methane emission, that means that the greenhouse gas emission will be reduced further. All right, now what are the technology that had been used for developing these varieties? So I told you the CRISPR-Cas system had been used. The CRISPR-Cas is nothing but it is the molecular scissor. All right, when you have to cut the DNA, DNA is a long strand of the genome, and if you have to, there is a long strand of genes, and if you have to cut those genes, so you require scissors. All right, if you have a cloth, and if you have to, cut the pieces of the cloth, you will require scissors. In the same manner, if we have to bifurcate, or we have to have a strand of the DNA that has to be cut, we require a specialized molecular scissor, and that is called as CRISPR-Cas. Right now, we are using CRISPR-Cas9. All right, then there are site-directed nucleases, or SDN. We are using two methods, SDN1 and SDN2, for the site-directed nucleases, where we make the desired changes and there are no foreign DNA that are introduced into these varieties. So over here you can see how this works. We have the CRISPR-Cas9 molecular scissors that are used for the cutting of the DNA, the faulty DNA or the faulty genome strand or the genes which are there. All right, it has been cut. After the cut, it is deleted, and then we have the SDN1 or SDN2 techniques which are used for the site directed the changes that have to be done, the modifications that have to be done, and later on we get the perfect genome in it. Now, what is the regulatory framework in India related to the genetically modified crops? So before 2022, the regulatory framework was a stringent framework that was there, and it required very strict regulations that were there. Very strict GM regulations were there before 2022. However, when we are talking about after 2022, there was an exemption that was made for SDN1 and SDN2 techniques. They were exempted from full GM or the genetically modified oversight committees. All right. Now, over here, when we are talking about the current status, biosafety evaluation and approval processes are basically underway for the SDN1 and SDN2. All right. Along with this, the regulatory shift had accelerated the genome edited crop development in India, but it raises certain questions. I told you that there are pros and cons for everything. Now, over here, there are certain issues that have to be dealt with. The first is the intellectual property and the seed sovereignty issue. When we are talking about CRISPR technology, the CRISPR technology is basically the patents are held by the institutes, like we have Broad Institute as well as UC Berkeley Institute, and that creates a complex licensing requirements. There is a licensing fees that has to be paid for using these patents, and this licensing cost will get burdened onto whom? This licensing cost will get burdened onto whom? Not to me or not to these companies or the institutes which are using it. Of course, the use of these licensing fees, the cost will increase. And when the cost will increase, that will increase the cost of the seeds, and ultimately the technology, the license fees that would be passed on to the farmers, all right, the farmers through the seed prices, and it will make the advanced varieties unaffordable for the small and the marginalized farmers. And when I'm talking about the small and marginalized farmers in India, they constitute about 75 to 80% of the farmers' population. All right, we have more of small and marginalized farmers. So in this manner, there would be about 15 to 20% of the population of the farmers or the farmer community that would have the access to these seeds. All right, if the price of these seeds will be more. I'm sorry. All right. Then comes the another issue, and that is the farmer seed rights. There had been traditional farmer practices for saving, exchanging and replanting seeds, which could be restricted by the intellectual property right patents or the the patents that would be there, and that will strengthen or that will threaten the agriculture practices in India. All right. Along with this, there is seed sovereignty concerns which are there, the dependence on the proprietary technologies which are there, controlled by the foreign entities like we have Berkeley Institute. It raises the questions about India's agriculture self-reliance and the farmers' autonomy over their own crops. So these are the different issues when it comes to intellectual property right as well as seed issues which are there. Along with this, we have data transparency concerns as well, because when we are talking about the data that had been published, all right, initially. So now there are gaps which are there. The gaps which have been noted in the published trial results. So there is incomplete data that is there. Along with this, there are selective sites which have been used for the field trials. Now, when we are talking about India as a whole, India does not have a same kind of or similar kind of ecosystem all throughout. We have different kind of ecosystems which are there in our country, right from the desert ecosystem to we have mangroves, we have the the coastal region where we have mangroves, we have the kind of ecosystem where we have the tropical climate that is there, we have the ecosystem where we have the temperate regions which are there. So of course we have different kind of ecosystem, and with the selective sites, all right, with the selective sites which are in limited geographical regions, these kind of trials are not for complete India. Sorry. All right. Now, when we are talking about why we are talking about this in today's session, because there had been discrepancies which have been noted in the field trials, all right, discrepancies which have been seen or claimed between the claim benefits of these two different varieties and the data that has been collected by AIC RPR, that is All India Coordinated Rice Improvement Project data. Both these datas mismatch with each other. All right. So when we are talking about the mismatch of the data, it is very important that there is a need of openness, and the the data that is there, the data sets which are there with the government or with the different organizations, they have to be comprehensive and also publicly accessible so that everyone could know about the benefits as well as the the losses that could be incurred upon using these different kinds of crops. All right, now when we are talking about the safety and the off-target effects of these genome-edited crops as well. Now, we are editing a genome. All right, we are using CRISPR-Cas9 technology, we are using SDN1 and SDN2. But what happens is that sometimes what happens is that we have certain intended things or mutations that we want to do. But at the same time, there are certain unintended mutations also that they accompany. All right, so over here we have intended edit, we have precise target change. All right, we have minimal predictable impact. But what happens if we had the unintended mutations? That will lead to off-target edits as well as unknown long-term effects. All right, so there would be unintended mutations that will happen. If there are unintended mutations, the changes will go beyond the targeted sites, which will lead to the changes in the metabolism. It will lead to changes in the allergenicity of the crops as well as the nutritional capacity of the crops. All right, along with this, the off-target edits, all right, they will change the genetic composition, and there would be the changes would be in the unexpected location, and we do not know what their long-term effects would be. And when we are talking about long-term effects, we are not talking about only about this generation, but multi-generational effects. When we are talking about the genome-edited crops or the genetically modified crops, the effects of these crops will not be only for this generation or for the next generation to come, but for the next 10, 20, 25, 30 or hundreds of generations to come. So it is very important that we have to think about the multi-generational effects as well while implementing these crops. Now, also, when we are talking about this, the two varieties of the rice that have been introduced, they would have export and market implication as well. When it comes to EU or European Union have a strict policy when it comes to the genetically modified crops. And the genetically modified crops, they have to be labeled as well as get approved before you are exporting it to the European Union. And when we are talking about this approval and labeling, so that will create the labeling issue again. We have to maintain separate supply chains for the genetically engineered or the genetically modified crops and also the traditional varieties. And also there is a problem of interbreeding, or there is a problem of risk contamination between these two varieties that could be there. All right. Apart from this, there are potential trade barriers for the genetically modified rice when it comes to the premium markets like, for example, Japan or Korea. They seek the non-genetically modified rice or the traditional rice. They are focused on the traditional rice more. And also, when we are talking about the gene pool diversity, that would be there because there would be genetic contamination of the traditional varieties because of the reduction in the genetic diversity. And again, that could be an issue, and that could lead to export and market implication in the near future. Now, when we talk about the other dimensions of introducing the genetically or genome-edited varieties of rice, it is the farmer's right or autonomy in the seed selection, the seeds choices, and the traditional knowledge protection which had to be looked forward and cared for. Apart from this, the seed access should be equal. I told you that the seed access should not be only to the farmers which have the access to monetary, or they are from well-to-do families. All right, we have more of small and marginalized farmers. They should also have equitable access to the seeds so that they could also reap into the economic benefits. And also the rice, which is used as akshat, also holds a cultural value in our system, Indian system. So therefore, it has to be seen that ethical, social and cultural dimensions of it should be looked after very well before introducing the varieties in India. All right. Now, when we are talking about the broader GM or genetically modified crops or landscape in India, we have different varieties of different crops which are currently into, they are into development phase. When we are talking about the oil seeds, we have the varieties of, under oilseeds, we have the varieties of mustard, GM mustard and the groundnut, which are basically right now, they are under development. Alright, GM mustard is basically awaiting the commercial approval, and we have groundnut and soybean crops which have been developed for the drought resistant varieties, and they are under the development phase. All right, when it comes to pulses, we have the varieties of chickpea and pigeon pea. Again, they are in development phase. All right, they are under development. Then we have cotton. Only when we are talking about the only commercialized GM crop in India is BT cotton, and it has been already approved in cotton. And in vegetables, we have brinjal and tomato varieties which are right now under the development phase. So these are the different varieties which are under development except from BT cotton when it comes to genetically modified or genome edited crops in India. Alright, now what are the advantages of introducing the GM crops in India? So they are climate resilient. I told you that they are adapted to go through drought conditions, heat as well as salinity issues. Alright, they provide with provide with the efficiency when it comes to the resource usage because they reduce sorry they reduce the water dependency as well as they reduce the nitrogen fertilizer usage as well. So that leads to resource efficiency as well. Alright, there is enhanced productivity on the same land area and that gives us better yields. And also when we are talking about it, they are tools for resource poor farmers and that leads to the support for these small and marginalized farmers in terms of better income, generational capacity as well. But they have risk and limitations as well. The first and foremost risk is, the loss of the genetic diversity. The loss of the genetic diversity because we will be over-reliant on fewer capacities or fewer varieties of the crops and that will have a risk of monoculture. Alright, another risk is that there would be a biodiversity loss because the traditional varieties and the wild relatives of these varieties, they will get endangered because we will be relying more on the traditional, on the GM crops while we will be cultivating them. There is the farmers, they are logged into the commercial seed system and that creates the technology dependency of these farmers. Also, the high seed prices because of the licensing fees that will include or that will lead to the increase in the cost prices. And again, when we are talking about the insufficient long-term monitoring and oversight in India, that could lead to the regulatory gaps in India. So these are the risks and limitations while introducing the GM crops in India. Now, this is not just about technology. When we are introducing the GM crops in India, we have to look towards the ecological system as well or ecosystem as well. We have to take into account the ecosystem perspective because when we are talking about single gene solution, there has to be a holistic view when taken into account. We do not want to risk monoculture in India. Alright, at the same time, the farmers should be taken into the center when it comes to the decision making. The decisions must prioritize their agriculture, this agriculture communities and their agriculture priorities, their traditional knowledge as well. And there has to be a rigorous, transparent scientific assessment that is required before introducing these crops in India. So, if the introduction of these crops, yes, they hold very prominent importance when it comes to the food security, but it has to be evidence-based. All right. Now, what are the policy recommendations for India? So, there has to be a transparent data disclosure when it comes to genetically modified or genome-edited crops. There has to be an independent biosafety institute that has to be, it has to be established as an autonomous body, which will be free from all the commercial interest. Alright, then there has to be a strong regulation where a robust oversight should be there, including post-release monitoring and not just the monitoring during the development phase. And also the farmer participation should be there in the trials so that the real testing, the real world testing could be done in the real-time manner. Alright, when we talk about the farmers and the small holders, there could be a roadmap. They have to be given affordable, equitable distribution mechanism when it comes to the technology. There have to be proper access to the technology that has to be provided to the farmers. Along with this, the training and education should be provided where we have most of our farmers that they rely on the traditional knowledge system when it comes to farming in India. All right. Along with this, there has to be a protection of the traditional seed saving practices and they have to be given, the chances to produce the seed themselves. And when we are talking about the ongoing technical assistance and resources that has to be provided, the support should be provided to the farmers and the small holders that are there. Now, this topic holds very importance for your UPSC. Why it holds very importance for your UPSC? Because When we are talking about GS paper 3, you can note down over here, we have agriculture where there are topics like biotechnology, crop improvement, food security. Alright, we have topics like gene editing, CRISPR application, innovation in science and tech, as well as we have biodiversity, biosafety, sustainable agriculture that are part of the environment. And when we are talking about the ethics, the farmers' rights, seed sovereignty and intergenerational equity forms the basis, or we can have the questions related to the ethics. And when we talk about the governance, we have regulatory frameworks, transparency, public policy, and in social issues, we have equity in technology access and cultural dimensions that should be there. So now, when you are answering any question based on these topics and based on today's session, so what you have to need or what you have to acknowledge while writing these questions. You have to acknowledge the potential that these genetically modified crops holds for India as a whole and for the globe as well. But at the same time, you have to be cautious. Alright, you have to be cautious. You have to take precautions while writing everything down. You have to be critically, you have to analyze critically the complete how the question has been asked and what all things you have to write there. Alright, you have to discuss the pros, but at the same time, the cons should be also developed in the answer. Alright, so that is the conclusion over here. Alright, and when we are talking about the promise of the gene edited or the genome editing crops, that holds a very good promise for the Indian agriculture, but it requires robust regulation along with this transparent science and farmer-centered implementation to balance the innovation with safety and equity. Now, you can practice these questions. You can try writing the answers for these questions. We have 3 different questions. Should India prioritize GE technology over conventional breeding programs? The second question is, how can we balance innovation with long-term biosafety and environment protection? And third question is, what safeguards ensure technology benefits smallholders, not just large agri-businesses? So you can try and write these 3 different questions, the answers to these questions. All right. So that would be all in today's session, and I hope that you have now understood the importance of today's session, why it holds very importance for your UPSC preparation. Please do not forget to like, share and subscribe to our channel. That would be all. Thank you. Bye-bye. Take care and Jai Hind.